Okay, so welcome to this next video in which we are discussing voltage-gated sodium channels. Okay, so we've just given the motivation for studying the voltage-gated sodium channels in detail, which is to study the interesting things that they do within an action potential. Okay, so we're going to start off by discussing the structure of the voltage-gated sodium channels and by discussing the many different types of voltage-gated sodium channels, because there is not just one type of voltage-gated sodium channel. They all have a similar structure, but there are many different types basically. So, basically voltage-gated sodium channels have two types of subunit. They have a subunit known as an alpha subunit, and then they can also have one or two beta subunits attached to that. Now we're going to start by discussing the alpha subunit, because the alpha subunit on its own is actually a functional voltage-gated sodium channel. So the beta subunits are just auxiliary subunits, and indeed you can have voltage-gated sodium channels which don't have the beta subunits. Okay, generally they do have them, but the actual pore-forming unit which opens in response to depolarization of the electrical potential difference and conducts sodium ions is the alpha subunit. So we're going to start off by looking at this alpha subunit of voltage-gated sodium channels. Okay, so alpha subunit. Right. Okay, so it is one protein is the first thing to say, and it's going to fold up to make the entire pore-forming unit of the voltage-gated sodium channel. So I'm actually going to start off with its membrane-spanning topology, and then we'll discuss its more gross structure of how it folds up to make the ion channel. Okay, right, so it starts off with its amino terminus in the cytoplasm, okay, as shown here. Then you have the first membrane-spanning alpha helix, then the second membrane-spanning alpha helix, then the third, then the fourth, and you better like membrane-spanning alpha helices because you're about to see 24 of the things. Right, so, then the fifth membrane-spanning alpha helix, then a p-loop, then the sixth membrane-spanning alpha helix. Now that's the end of a structure known as domain 1. Okay, so D1 stands for domain 1. Now you repeat this same structure over again. So you have a first membrane-spanning alpha helix, a second membrane-spanning alpha helix, a third, a fourth, a fifth, a p-loop, and then a sixth. And that then is now domain 2. Okay, and you can see that it's got the same structure as domain 1, hopefully. Now you're going to have a loop again between domain 2 and the start of domain 3. Okay, so there's our first and our second membrane-spanning alpha helices, our third, our fourth, our fifth, our p-loop, and then the sixth membrane-spanning alpha helix. So the thing that I'm calling the p-loop is this re-entrant loop here, where it goes into the membrane and then comes back out again. Okay, so this is known as the p-loop, and it's called the p-loop because that P stands for pore, and we're going to see that this loop is actually going to line the pore at a portion of the voltage-gated sodium channel. Okay, so more of that later. Okay, then we finally, oh by the way, this is called domain 3 here, so we've now completed domain 3, and then finally we have domain 4. So again, here are our 5 first membrane-spanning alpha helices, our p-loop, and then our sixth membrane-spanning alpha helix, like so. And then this is our carboxylic acid terminus. So overall we've got 24 membrane-spanning alpha helices here, but it's not as bad as it sounds because we're really just repeating one structure 4 times. Okay, right. So, let's now give the naming system for these membrane-spanning alpha helices then. So let me draw a bigger picture where we zoom in on one of these domains. Okay, so I'm just going to take a domain out now, and we're going to name up the membrane-spanning alpha helices. So 1, 2, 3, 4, 5, the p-loop, and then our sixth one here. Okay, right, so the first membrane-spanning alpha helix of each domain is called S1. The second membrane-spanning alpha helix is called S2. Okay, the third membrane spanning alpha helix is called S3. This fourth membrane spanning alpha helix is called S4. The fifth is called S5. And then we have the P loop here, the pore loop. Okay, and then finally we have the sixth membrane spanning alpha helix, which is called S6. So if you want to label up a membrane spanning alpha helix on this structure, you tell me which domain it's in, and then you tell me which membrane spanning alpha helix it is within that domain. So for instance, if I take this one here, we're in domain 3, and I'm pointing to S4 of domain 3, so I call this domain 3 S4, so D3 S4. Okay, right. So that's the membrane spanning topology of these alpha subunits. Okay, and there isn't just one alpha subunit, there are many different ones, but they all have this same membrane spanning alpha helix. Okay, and basically what's going to happen is this protein is going to fold up and produce the voltage gated ion channel. Okay, so let me show you. So, in reality, this structure is going to fold into a shape that I can draw like so. Okay, so here's our nice cylindrical ion channel here. Okay, sitting in the phospholipid bilayer. So this represents the outer leaflet of the phospholipid bilayer, and this represents the inner leaflet. It's a bit late to say that after I've used that in all of these pictures, but never mind. Okay, and then here is the ion pore down the middle. So basically, the four domains are going to make the 4/4 of the sides of this ion channel then. Okay, so this one at the front here, this is domain 1. So this quarter here, this is made up of this portion. Then this next quarter will be made up of domain 2, this portion here. The next portion will then be made up of domain 3, this portion over here. And then finally, this portion here will be made up of domain 4. So we're going to wrap this thing round to form this ion channel like so. Okay, right. So now what I want to do is come back to this concept of the P-loop lining the pore, basically. So if we look at this channel from above, so let me draw another picture where we look at the ion channel from above. So we're going to look down like so. Okay, and this is what we'll see. Okay, so we're looking from the extracellular fluid, and here is the channel through which the ions move, right at the center there. Okay, and here are the 4 domains that make up the 4/4 of these voltage-gated sodium channels. So we have domain 1 here, we have domain 2 here, we have domain 3 here, and we have domain 4 here. Okay, like so. Basically, the portion of each domain that is closest to the pore here, at least on the extracellular side of the channel, is the P-loop. Okay, so this portion here, this little inner ring here, this is made up of the P-loops. Okay, so I'll colour them in a certain colour. So let's have them in red here. Okay, and basically this is the same as this portion here. So this is the P-loop of domain 1, so this is this portion in domain 1. Here is the P-loop in domain 2, so that's this portion here. This is the P-loop in domain 3, so that's this portion here. And this is the P-loop in domain 4, so that's that portion there. Okay, now of course the P-loops don't span the entire way down the pore, so this isn't going to be making up the pore the entire way through, because it can't, because this doesn't go all the way through the membrane. So you're only going to have this top portion at the top, basically, at the extracellular portion. And you'll be able to see that more when I draw another picture in a moment. Okay, so the other crucial domain that we need to discuss is this S4 membrane spanning alpha helix of every single one of these domains here. Okay, S4 is extremely important. Okay, so S4 is actually the voltage sensor. So it is this membrane spanning alpha helix which is responsible for sensing the depolarization of the electrical potential difference across the cell membrane and opening in response to that depolarization. So this is the voltage sensor. Now how does it do this? Well, basically if we draw S4 out here, We'll just represent it as a rectangle here. And we've got 4 of them, of course. We've got an S4 in each one of the quarters of the alpha subunit. Okay, basically S4 has loads of positively charged amino acids within it. So it's a membrane-spanning alpha helix, and actually every third amino acid is positively charged. So what does that mean? That means that if I was to stretch out the polypeptide that is wrapped around in an alpha helix here, and I was to look at the amino acids one by one, I'd find that every third amino acid has a positive charge. So let's say this one here had a positive charge. Then I'd count along again, 1, 2, and then the third one would have a positive charge. Okay, and then again, I'd count along 1, 2, and then the third one would have a positive charge. So every third amino acid within this membrane-spanning alpha helix of S4 here has a positive charge, which means that the whole membrane-spanning alpha helix is very positively charged. That is what's going to respond to the change in the electrical potential difference, and it is going to cause the opening of the channel, but we'll discuss that more later. Okay, right. So the S4 membrane-spanning alpha helices will sit further back, so they'll sit like so. Okay, so here are the S4 membrane-spanning alpha helices, all 4 of them. Okay, and we'll come back to this picture later, because later when we come to discuss the opening, what we'll do is we'll take a cross-section through like so. So we'll cut down there, and then sort of cut the channel in half, and then look at what we see from the inside, basically. So we'll look from this sort of an aspect. We'll take away this first half, move that out of the way, and have a look from the side at what we'll see. So basically, we're cutting the channel in half, like so, taking this bit out of the way and having a look at what we see. And we'll look at that in more detail later when we discuss the opening. Okay, for now, what I want to talk about is all of the different alpha subunits that you have within, which are all capable of making a voltage-gated sodium channel. Okay, so there is not just one alpha subunit of a voltage-gated sodium channel is what I need to stress. It would be very, very simple if there was just one alpha subunit, but it's not that way. Instead, there are 9 different alpha subunits of voltage-gated sodium channels. Now, these will all have slightly different primary sequences of amino acids, so that primary sequence of amino acids will all differ. Okay, however, they all have this same membrane-spanning topology like so. They all have these conserved things like S4 being the voltage sensor and the P loops lining the pore. They all have the same structure. They just have different sequences of amino acids. Okay, and they do have slightly different expression patterns. So let's now discuss the different alpha subunits of voltage-gated sodium channels. So as I say, there are 9 different alpha subunits, okay, and they are called the NAVs. So you have NAV, and this stands for, Na is for sodium, the V is for voltage-gated, so this means the voltage-gated sodium channel alpha subunit, and then we've got 1.1, okay, we've then got NAV 1.2, we've then got NAV 1.3, and their naming is absolutely beautiful, basically. It goes all the way on to NAV 1.9. So you have NAV 1.4, NAV 1.5, NAV 1.6, NAV 1.7, NAV 1.8, and then finally NAV 1.9. So that's the simple naming system for all of these alpha subunits of voltage-gated sodium channels. Okay, now, you find these different alpha subunits in different excitable cell types. So remember, the only cells which will have voltage-gated sodium channels are excitable cells, cells which can fire action potentials. And the main examples of cells which can fire action potentials are neurons and also myocytes, muscle cells of both skeletal and cardiac type. So, basically, these alpha subunits of voltage-gated sodium channels have different expression patterns. So what you find is that within the central nervous system, you have mainly NAV 1.1 alpha subunits, NAV 1.2 alpha subunits, NAV 1.3 alpha subunits, and also NAV 1.6 alpha subunits. So these are the sorts of alpha subunits of voltage-gated sodium channels which you will find in the central nervous system. Okay, so in neurons within the central nervous system, these are the sort of alpha subunits that you have. Okay, whilst in the peripheral nervous system, the alpha subunits of voltage-gated sodium channels that you find are NAV 1.7, NAV 1.8, and NAV 1.9. So these three are the forms that you find within the peripheral nervous system. Okay, and then finally let's discuss the ones which are in muscle cells. So NAV 1.4 is found within skeletal muscle. Okay, so this is in skeletal muscle. And then finally the one remaining, which is NAV 1.5, is in cardiac muscle. Okay, so NAV 1.5 you find within cardiac muscle. Okay, so those are the 9 different alpha subunits of voltage-gated sodium channels. And we've now seen the different expressions of them. So NAV 1.1, NAV 1.2, NAV 1.3, and NAV 1.6 are found in central nervous system neurons. NAV 1.7, NAV 1.8, and NAV 1.9 are found in peripheral nervous system neurons. And together these 7 are therefore called the neuronal type alpha subunits of voltage-gated sodium channels. And then NAV 1.4 is found in skeletal muscle cells, and NAV 1.5 is found in cardiac muscle cells. Okay, right, so those are the alpha subunits of voltage-gated sodium channels now covered. Let's now turn our attention to the auxiliary subunits. Okay, so these are the beta subunits, which you can join onto the alpha subunits of your voltage-gated sodium channels. Now, I want to stress that you don't need the auxiliary subunits. They are not responsible for the conduction of sodium ions through the pore, and they are not responsible for the voltage gating either. The alpha subunit on its own is a perfectly functional voltage-gated sodium channel. It will open in response to depolarization of the electrical potential difference across the cell membrane, and it will allow sodium ions to move through it. The auxiliary subunits combined to the outer aspect of the voltage-gated sodium channel alpha subunit, and they can moderate the properties of it to an extent, and they can also guide it to certain portions of a cell's membrane, basically. So the function of the auxiliary subunits is mainly to control where the voltage-gated sodium channels are actually positioned within the cell. Okay, so basically there are 4 different auxiliary subunits, and they're known as the beta subunits. So we've got alpha subunits and beta subunits. Okay, so there are 4 different beta subunits, and they are called the NAV beta 1, okay, through NAV beta 4. So again, it's a very simple naming system. So you have NAV beta 1, NAV beta 2, NAV beta 3, NAV beta 4. Okay, right. So let's have a look at the membrane spanning topology of these beta subunits. So they have a very simple membrane spanning topology. Okay, so if this is the outer leaflet of the phospholipid bilayer, let this be the inner leaflet of the phospholipid bilayer. And basically these beta subunits have their amino terminus extracellularly, and then they have their carboxylic acid terminus intracellularly, and they span the membrane a single time. So they have a single membrane spanning alpha helix. So they have an amino terminus, a transmembrane domain here, and then a carboxylic acid terminal domain over here. Okay, right. Now, basically, alpha subunits can either have no beta subunits attached to them, or they can have 1, or they can have maximum of 2. So I now want to explain to you the rules by which you can attach beta subunits onto alpha subunits. So let's draw our alpha subunit here. Okay, so we know that there are now 9 different alpha subunits of voltage-gated sodium channels, which are found in different sorts of excitable cells. Okay, and which do have slightly different properties because of their slightly different amino acid sequences, but they all have the same membrane-spanning topology, and they're all activated by depolarization. So here's domain 1, here back here is domain 2, over here is domain 3, and then here is domain 4. Okay, now, basically, to domain 1, you can attach covalently either a beta 2 subunit or a beta 4 subunit. So over here, near domain 1, you can attach either beta 2 or beta 4. So you have to pick one of them. You can pick either beta 2 or beta 4, or you can pick neither. You can say, I don't want to attach one of those on. Okay? And if you do choose to attach it on, it's attached covalently. Okay? So by a strong covalent bond to this number 1 domain here. Okay? There is then the option to add another beta subunit onto domain 4. Okay? So over here, and this can either be beta 1 or beta 3, okay, or none again. And if you attach one of these onto domain 4, it will be attached ionically, or at least non-covalently. I don't actually know that it's ionic, but non-covalently. Okay, so this interaction is non-covalent, and this interaction is covalent. Right, so you start off with your alpha subunit, and when you're picking your auxiliary subunits, you've got a maximum of 2, and you can have none if you want. So you have to decide, do you want one of these covalent ones attached to you? If so, which one do you want? Either beta 2 or beta 3. And then you have to decide, do you want one of these non-covalent ones attached to you? And you can choose either beta 1 or beta 3. Okay, right. So that then now is the structure of voltage-gated sodium channels complete. What we'll do now is take a break, and then after the break, what we'll do is discuss the opening of voltage-gated sodium channels. Okay, so how does a depolarization in the electrical potential difference across the cell membrane trigger a voltage-gated sodium channel to open?